students. I want to thank Dr. Frazier for being here. I want to thank Ms. Randallette Williams for helping to facilitate getting this going, and all of our awesome USG staff as well, Melissa Gattuso and Cheryl Thomas, who are on the call, who made it possible for you guys not only to be here and get your wonderful supplies that you receive, but also to get your Grubhub $15 food gift certificate for being here as well. So, Cheryl, did you want to say anything really briefly about that process before we get started?
Yes, if you have already have a Grubhub account, the instructions you received would help you to link your account to this event. And so at 6:15, you won't see that $15 credit until 6:15. That will be in your account when you check out. So if you select the item that is, exceeds the $15, you would need a debit or credit card to pay the balance. But Gear Up has given you a $15 credit this evening to purchase your dinner meal. Was everyone able to connect, follow the instructions this afternoon?
Yes, ma'am.
Okay. So at 6:15, that credit will go live. And thank you for joining us tonight.
Thank you, Cheryl. We're going to hand it over to Dr. Frazier.
All right. Well, hi everyone. I'm playing around with scenes, by the way. I just always do that on Teams. I have no idea why, but I'll like put people in all sorts of things. But at any rate, so the students that I have, yeah, I know I talked to Stephanie earlier. Daisy is on, correct? Is Daisy here? Maybe not.
Yes, she's here.
Oh, okay. Daisy, I just wanted to get a quick introduction. So are you in high school, middle school? If high school, what grade are you in? And what are your goals?
I'm in high school. I go to Jurgen High School, and I plan on majoring in nursing.
And what grade are you in?
I'm going to be a senior.
Oh, another senior. All right. All right. Let me see. I have, is it Janae?
Yes, that's how you say it.
Okay. And what grade are you in and what are your goals?
I'll be a freshman in college in August.
Okay, awesome. What school are you gonna be at?
Savannah State University. Is the camera
Okay,
like kind of
awesome.
crooked or something?
You're crooked. I'm like, okay, what happened to you?
Is this right?
Do it. Yes, right there.
Okay.
Yeah, there you go. There you go. I'm like, oh my gosh, what happened to her?
That's my phone.
No, you're fine. You are absolutely fine. Let me see. So, and then I'm trying to think. What's the other one? Chastney. I think. No, I didn't talk to you already. What grade are you in?
Can you hear me?
Yes.
Okay, I'm going to the 12th grade and I plan on majoring in nursing as well.
Okay, awesome. Let me see. And was that everybody? It was just four, right? Daisy, Janae, Stephanie, and Chastney, correct? All right, perfecto. So anyway, so thank you all for coming. So it's all exciting. I know for three of you it's super exciting because you're going to your senior year. And this senior year promises to be a good senior year. So you will get your prom, you will get your football games, homecoming, graduation, all of that stuff, right? And Janae, you're just getting started, right? So you're like starting back over again, kind of at the bottom of the tier, if you will, and work your way back up to the top again in the school part. But thank you all so much for coming. I am Dr. Monica Frazier. I'm the chair of the biology department at Columbus State University. I'm also the director of the Southwest Georgia LSM Alliance. And it's an alliance where we try to get more minorities to go to graduate school and to get STEM careers. So that's what our program is about. And so we've got several schools that are a part of our alliance. Is Columbus State, Columbus Tech, Valdosta State University, ABAC, or Abraham Baldwin Agricultural College, and Georgia Southwestern State University. And soon to be, I guess I can, well, I don't know, I won't make that announcement. I'll hold off on that one. But at any rate, but all of those schools are part of our alliance. So if any of you are attending any of those schools, definitely consider joining the Lewis Stokes Alliance for Minority Participation. Janae, I know that you are going to Savannah State, but I believe Savannah State has a LSAMP program as well for you to be a part of if you would like to be a part of that. Okay. All right, so I am going to share my screen and hello, come on. Okay. So all right, so this is an ELIZA lab with Gear Up Georgia Biotech Career Exploration for summer 2021. Again, I'm Dr. Frazier. And what I want to do is I want to start this session off with an introduction on viruses. Viruses is actually something that I have my PhD in. So I'm a virologist by training. And so I study a lot of viruses actually, and I find them very fun and very exciting. But viruses are essentially just small particles consisting of essentially two things: a nucleic acid that's surrounded by a protein that we call a coat. Now, when we talk about nucleic acids, what are the different types of nucleic acids? Who remembers the types of nucleic acids? And there's two, just to give you a hint. Anybody remember?
Did they begin with an A, like both?
Huh?
Did they begin with an A, like both of them, or?
One of them begins with the letter D, as in dog. And they have a letter A at the end on both of them. And it's 3 letters. So when we look at nucleic acids, there are 2 types of nucleic acids. You have your DNAs and you have your RNAs. Anybody remember those? And I can't see you, by the way, so you'll have to speak because I'm presenting, so I can't actually see you at all. All right, yes. So you will have, viruses are composed of either an RNA nucleic acid or a DNA nucleic acid that's actually surrounded by a protein coat. And so what you see here on the right is actually a picture of coronavirus. So you don't see the nucleic acid on the inside, but what you see are all of the proteins that make up the coat that's actually enclosing all of this. And so this is what the coronavirus looks like. And one of the things that is unique, this is a cross section of the coronavirus, the one that we're dealing with or have been dealing with for the longest, the SARS-CoV-2. And so on the inside is the nucleic acid, which is represented in blue. For coronaviruses, the nucleic acid, remember I said there are 2 different types. And what were the 2 different types again?
DNA and RNA.
Right, DNA and RNA. For coronavirus, that nucleic acid is RNA. So because the genome is made up of RNA, we call them RNA viruses. Okay? And RNA viruses are really unique in that they have a tendency to change and mutate a lot and quickly, unlike their DNA virus counterparts. But as you can see in this picture, you can see the nucleic acid in blue, which is here, the RNA, and it's surrounded with a coat, just like if we're outside and it's cold and you put a coat on. So that particular coat consists of all of these proteins. There's membrane proteins, spike proteins, envelope proteins, nucleoproteins. All of these proteins come together to make a coat. Now, the membrane protein, I would call that the base of the coat. Okay, so you know you got your fabric that your coat is actually made out of. It's got sleeves and all this stuff. But then sometimes you have some embellishments on your coat, right? You might have buttons on your coat. You may have some hooks on your coat. Any little thing that kind of adds a little attraction or something to it. I'm going to refer to those things as your spike proteins. The spike proteins are those things that are on the surface that kind of gives it some sort of an identity. And it's actually the spike protein that your vaccines are actually targeting. So that when the vaccine that you receive, they're actually made to recognize this particular spike protein. Now, you'll notice over here on the left, I have on here SARS outbreak 2003. Something, just a unique fact here is that coronaviruses aren't new. They came on the scene in the news and everybody was like, oh my goodness, this coronavirus. And of course, I'm sitting there thinking, oh yeah, I remember that one. I remember the whole family. Because viruses are a part of a family, and coronavirus is the family name, if you will. But there was another coronavirus outbreak in 2003. So anybody know what you were doing in 2003? And for those of you on the call, were you even born yet?
No.
If it was like
No.
after June, yeah, I was born.
You were born if it was after June? Well, I can tell you it was definitely after June in 2003. It was definitely after June. It actually happened sometime around in the fall, winter months. There was a huge outbreak, and this is a picture of the Newsweek magazine. And you'll see here, what does it say? SARS, can it be stopped? The new age of epidemics. And you'll notice the gentleman, what is he wearing?
A mask.
A mask. And so everybody, you know, so for me, it was kind of, okay, this is what's going to happen, right? But you'll notice that in the US, that wasn't, it was kind of different here. Had you ever heard of a SARS outbreak before or a coronavirus outbreak before? Anybody ever said anything about there being an outbreak of coronavirus in the past?
No.
No? Hadn't heard about it,
I heard
huh?
of it being one in 2019. Like they said it started in 2019. Like that's why they
Okay.
said COVID-19, you know?
Right. Mm-hmm. Right. And so what I want to do is just kind of let you all know that this particular virus, having an outbreak such as it was not anything new. But do you know why you probably never heard about it? And I can tell you why. The reason is it never made it to the US. It made it to Canada, but it didn't make it to the US. And the reason, another reason why I'm like so familiar with it, besides the fact that I happen to be a virologist, is because I was actually headed to a virology conference in Canada in 2003 when that outbreak occurred. And I ended up not being able to go to the conference because Canada stopped everyone from traveling into their borders or out of their borders. And they didn't want anyone coming in or out of the country. And so when they closed down all of the borders, what happens is the virus eventually died off. Everybody was wearing masks. Everybody just kind of stayed away. Everybody just went away. And so SARS never actually came to the US. And so therefore, many people in the US never even heard of coronavirus or even knew it existed. Pretty interesting, huh? All right, so let's look at ELISAs. Okay, so what are ELISAs? ELISA stands for Enzyme-Linked Immunosorbent Assay. A whole lot of something to when we just say ELISA. We don't even say all of that in terms of the terms. But what they are assays that are actually used to diagnose diseases. And the way they do it is they use a field in biology called immunology. And in immunology, let me ask this, has anyone ever heard of immunology, or what do you know about immunology? Anybody know anything about immunology?
I've never heard about that.
No? When you think of the word, and I'm going to spell it here. When you look at this word, what comes to mind?
Is it the study of being immune?
Right.
Like,
it's like your
okay.
immune system. And what do we know about the immune system?
It fights off diseases like with white antibodies to like protect your body from getting sick or whatever.
Exactly. And that's what this particular assay does. It actually is a immunological assay that actually looks for antibodies and antigens in a substance, and then you can actually detect the presence of those things. And of course, if you have antibodies, then that means that you're actually fighting off something, right? And so if you're producing an antibody against something, that means I'm fighting something foreign in my body that shouldn't be there. And these particular assays can actually detect the presence of those antibodies and say, yes, you have this particular agent, whatever that is, that's inside of your body. So in this particular instance, when the case of COVID-19, what you're measuring is an antibody response to the coronavirus, such that if you're infected with the coronavirus, because coronavirus is not native to your body, then what happens is your body recognizes that as something foreign. And because it's foreign, your body says, hey, you don't belong here. And so your body's immune response will then kick in. You'll start to produce antibodies. Those antibodies will attach themselves to the virus. And once they attach themselves to the virus, your body can recognize that and then destroy it. And so what you see here on the right is a COVID-19 cassette. This is an actual cassette that people can use. And what you do is where it says the letter S here, you put your sample there. And as you put your little sample in there, what's going to happen is you're going to see some lines appear on the screen. And if those lines appear, then that says that you're positive for COVID-19. If they don't appear, then you're negative for COVID-19. Anybody seen something like this before? All right, so that's pretty much what the ELISA is. Now, what I want to do now is show you a picture of the ELISA assay. And so you see here it says it works by immobilizing an antigen or antibody to a surface and then applying the sample to allow binding between the antigen and the antibody for development of an antigen-antibody complex. A lot of words, but let me just simplify it. This particular thing here is the surface, okay? And the surface can be anything. In this particular case, it would be to this plastic here, okay? Or I should say the plastic here, okay? So that's the surface. And what you do is you apply an antigen or an antibody to this particular surface. Now, what's the difference between the antigen and the antibody? The antigen would be the actual substance or particle that you're trying to detect. So in the case of coronavirus, and this is my pitiful little virus over here, we're going to have viral parts. And if you remember when I first started, I said that the vaccine acts against a particular part of the virus. And that particular part is the spike protein. So for the assay, the spike protein would be your antigen, okay? So in this particular instance, the antigen could be my spike protein. Okay. So I will take that spike protein and I will immobilize it, meaning I'm going to attach it to a surface. Whether I glue it down like you would a piece of paper or something, but you're going to attach it and affix it to a surface so that it doesn't go anywhere. It's immobilized. Then what happens is you have an antibody, and you add an antibody to this particular system that's going to recognize the antigen. In this particular case, we're talking about an antibody that can recognize the spike protein. And when it recognizes it, what it does is it will bind to it, just as you see here. So you'll see the binding interaction between here. But the problem is that, yeah, so the antibody attached itself to this particular spike protein, but I can't see it. I need to be able to see it. And if you remember in this assay over here, I said that if it's positive, you'll see a line show up. That's something I can see. So I've got to be able to detect the binding of the antibody to the antigen. So what you do is you have a secondary antibody, okay? You take a second antibody, and what's unique and special about this second antibody is not only will it recognize the first one, but attached to it is something that will produce a color or emit light, such that when the second one binds to the first one, and you've got this nice little sandwich here, when that binding interaction occurs, this particular guy is going to react. So either a color will be produced or you will see it light up, okay? And so in this particular case, what you'll see is a color produced, and that color will produce a band in the middle. Any questions on that? I'm going to come back over here to you guys. And
No, I don't have any questions.
all right, are you guys still looking at my screen, or? I think so. Yeah. Okay. Sorry. All right. So does that seem simple enough?
I had a question for you, Dr. Frazier, really quickly. Is this the same technology used in other tests, like pregnancy tests and stuff like that, to change that line?
Exact same one for the pregnancy test, exact same one for the drug test that you purchase in the store, exact same technology. Exact same, not different at all. Okay. Any other questions? Everybody pretty clear on how this thing works? How we just bind one on top of the other, on top of the others, almost like Legos, right? We're just attaching one on top of the other until we get to the one where it's going to give us a sign or a signal that these interactions occurred, okay? All right, and so that's pretty much what the ELISA is. And so what we're going to do tonight is we're going to do a simulation of a COVID test. This is the same sort of COVID test that people were taking all the time, and some people are still taking before they go on a trip. We're going to simulate what that's like. Okay, so this is the part of the packet. So you should have this that came in your kit. And I just want to make sure everybody has everything. You should have a little card. Whoops. I need going too far in. You should have a card like this. We're going to need to make sure we have that card out. So if you want to prop it against something, I know you guys also have a cup of some sort. So maybe you can prop it against your cup. Something else you should have.
I'm having a hard time.
I'm sorry, what's that?
Sorry.
No, go
I'm
ahead.
having a hard time propping my phone up somewhere. It won't stay. Oh, there you go. Okay.
There you go. You got it. You got it. I know you got those skills, girl. Perfect. All right. Then we've got our micropipetter, syringe micropipetter. You should have that. You should also have this microtiter plate strip. I don't have these, but you guys have them, but you have a lot of small little tubes that has a lot of words written on it, right? So you should have a total of, and I'm going to pull out my little instructions here. 1, 2, 3, 4, 5, 6, 7. You should have 7 tubes with all sorts of things in them. You've got your tips here. And of course, your gloves. All right, anything I missed? You got your cups, right? All right, so one of the first things that you do in science, one of the first skills you have to learn is how to micropipette. How to micropipette. Micropipetting is something that we do a lot in the lab. Now, let me say this. When it comes to measurements, right? If you want to measure something, a lot of times when you measure something, you say it's so many inches or feet. If it's a liquid, you say it's what, like 8 ounces of liquid, right? Or a cup or a pint, a gallon. Those are the terms that we use, correct? Well, in science, we don't use those terms at all. We use the metric system, and this is a system that every other person in the world, in the country, use except for us. But in using those, for liquids, we use liters, okay? And we use different variations of liters. Now, you may remember a long time ago in elementary school, you had to learn these different levels. You had to learn milli and centi and kilo and all of that stuff, right? And you probably were wondering, why am I ever learning this? I will never, ever use it in my life. And I hate to tell you, but in science, we use them all the time, all the time. And so this particular guy right here, it measures in what we call microliters, which is 10 to the negative 6 or 10 to the minus 3, depending on how many microliters we have. But we're measuring in microliters here, okay? And so on the card that you have, you'll see some numbers. At the very top, you see 200, then there's 100, 50, 20, and 10. These all represent microliters. So the 200 is 200 microliters, then 100 microliters, 50 microliters, so on and so forth, okay? So to use your pipetter, what you're gonna do is you're gonna take your tip, right? You're gonna take your tip and you're gonna attach it to the bottom of your pipetter, okay? And you want to make sure it's on there real snug because you want to make sure there's a nice tight seal there. Perfect, Janae. I like that. You did it. Okay. Now, this part at the top is like your plunger. So if you take your thumb and push up, you're not going to be able to push up very far, but it'll go up. You take your thumb, push it down, and you should be able to go up and down. Everybody feel that? Okay, so if you want to take up any liquid, what you're going to do is you're going to push up. If you want to dispense the liquid, you're going to push it down. Okay, so if everybody could, let's see, let's use the, does anyone have, can you guys go and put some water in your cup right quick? And not a lot of water, just a little water, because we're talking microliters here, so we're not even talking a lot. Wait till everybody get back. Yeah, that's perfect. That's good, Janae. That works. That works. Okay. So what I want you to do is I want you to take your pipetter and the tip, you want it to go just below the meniscus of the water, the level of the water. So you want it to go just inside of the water. And then what you want to do is you want to take your thumb and you want to push up on the plunger at the top. And when you push up on it, what I want you to do is take it out of the water and notice that you've got liquid in your tip. Everybody see liquid in the tip? Janae, you like, is that water in there? I don't see anything. Everybody sees water in the tip. All right. So without putting the tip in the water, what I want you to do is put it high above the cup, and I want you to press down and dispense it in there so you can see that there really is water in there. Oh, you got it now, right, Janae? Because she was like, I don't see anything in here. All right. Now what I want you to do is I want you to take up water again, do it again. But what I want you to do is the next time you do it, I want you to look at where the line is for the water, and I want you to compare it to the card and see how much is in your tip. Is it 200 microliters, 100 microliters? How much is in there?
Mine is in between 50 and 100.
Okay, what about you, Daisy?
50.
50? Chastney? What do you have?
100.
Okay, 100. And Stephanie, what about you?
Between 50 and 100.
All right, between 50 and 100. Now what I want you to do is I want you to dispense that out, and I want everybody to take up 50 microliters. Part of being a scientist and using a pipetter is all about accuracy. So if you're pipetting a certain volume, you want to make sure you're accurate and you're pipetting exactly that amount, not more, not less. You got it? Not so bad, right? All right, Chastney, did you get it? All right, Stephanie, we're good. All right, Daisy, we're good. Daisy, you still working on it? Oh, okay. All right. All right. And so what I want to do is I just want you to see how you have to be accurate, right? So you pipetted 50 microliters, you pipetted 100, and so on and so forth, okay? So now that we have pipetting down, let's actually go to the instructions on the instruction sheet and let's get started on the assay, okay? So you can go ahead and dispense your liquid out back into the cup, okay? All right. So the first part of the assay, what are we going to do? What's step number 1?
I don't have my paper. I thought I did. Hold on, let me go get it.
Oh, okay. That's okay. Daisy, you have yours? Yes. Okay. And what about you, Chastney and Stephanie? You got yours? All right. So very first thing is you're going to label the bottom of the wells. And so you see a picture here in the booklet with how you are to number them. So you're going to number the very bottom, not the top, but the actual back side of it. Okay. So if I hold it like this, it'll be the underside. Okay. And so you'll number them just like you have it there. It's easier to use a Sharpie. You can use a magic marker, but it's easier to use a Sharpie. All right, Janae, you got it. So what we're doing right now, Janae, is the first part, which is the labeling part. So we're going to label it first. And then what we want to do is on our activity sheet. So on the activity sheet, which is the second page of the activity sheet, what you want to do is you want to identify which sample is going where. Now, in terms of samples, which samples do we have? So for samples, you should have 4 tubes. The samples are the negative control. So if you can grab your negative control tube, your positive control, And they're all written on the tube. They're really teeny tiny writing, but they're written on the tube. It should say negative control, positive control, patient 1, and patient 2. Those are your 4 samples, okay? And what I want you to do is I want you to decide which one is going to go in well number 1, which is going to be for well number 2, which is going to be for number 3, and which is going to be for number 4. So out of those 4, negative control, positive control, patient 1, and patient 2, decide who's going to be 1, 2, 3, and 4. And so on your sample sheet that came in your kit, you should have a piece of paper that looks like this. Whoops. Me and this camera is just not having it today. Jeez whiz. You should see on there's a table that says well number 1, 2, 3, and 4. Everybody sees that? And so you just identify which sample are you using. The negative, positive, patient 1, patient 2. Once you have that identified and you know who you're putting where, at that particular point, I want you to take your micropipetter here, and we're going to pipette 50 microliters of the anti-human SARS-CoV IgG and IgM antibody into all 8 of the wells, all 8. So remember, we labeled them 1 through 4, and you got 2 columns of 1 through 4. So in all 8 wells, 1, 1, 2, 2, 3, 3, and 4, 4, you're going to put 50 microliters of the SARS-CoV-2 IgG, IgM antibody into all of them. And just to make sure we all have the right tube, everybody hold up your tube of antibody that you're using. Perfect. All right, Stephanie, Daisy, you got the same one? Yep. All right, perfect. Just want to make sure we got the right one. All right, so we're going to do 50 microliters. So we've already practiced how to pipette 50 microliters. So let's pipette 50 microliters into all 8. And you can use the same tip that you have here for all of them. Okay. Everybody's good so far? Excellent. When you're done pipetting 50 microliters in all 8, what I want you to do is take that tip off and you're going to throw it into that cup that you have water in. Okay? Into your cup of water. And then what you'll do is you'll get a new tip and put it on there. Now, once you have that on there, we're going to let it sit for 5 minutes at room temp, just on your table. Just let it sit for 5 minutes. So if you will, take your cell phones and set a timer for 5 minutes, and we'll let it count down. Okay? So if you can let it sit for 5 minutes. So Stephanie, are you done pipetting? Okay. Chastity, what about you? Are you done? No, not yet. Okay. Daisy, are you done?
Do we just put a drop in there?
So you'll take 50 microliters. Remember how we practiced taking up 50 microliters?
Yeah.
With the pipetter? So you're going to practice, take that same volume and you're going to put it in every well, 50 microliters.
Oh, so 50 microliters in every one?
Exactly.
Okay.
Mm-hmm. Did I lose somebody? I feel like I lost somebody. I lost, who did I lose?
Janae.
Oh, no, Janae. Okay, you're back. Okay.
My bad. My phone was telling me that I had 20% left.
Oh, okay. Did you plug it up? Oh, okay. We're going to hope and pray that you make it, right? All right. So, Janae, are you done pipetting? Okay. So while we're waiting these 5 minutes, what are your thoughts so far?
I feel like I'm working in a lab.
Yeah, very good. I will say I used this same experiment for my college students, and they felt the same way. They absolutely loved it. So it was fun for them. And to have the little pipetter to use. And so just realize that, has anyone had a COVID test done before or know somebody that had one done before? What, do you know what they did, how the test was done?
They swabbed your nose. They don't
Right.
hurt too.
What, they were like touching your sinuses too, right? Because they were trying to go all up in the sinus, right? It's like all up in your eye. You're like, really? Do you have to go that far? But anyways, but yeah. And so when they go in your nose like that and they pull the sample out for this assay that we're using, was that the antigen or was that the antibody? What do you think they were looking for?
What is it? It say, dang, what?
Or they
Anti-human. So I think it was bad.
No, do you think they were using the antigen or the antibody when they went in your nose and took the sample?
Antibody.
You think it was the antibody? Yeah, no, it was the other one. It was the antigen. Because what they're looking for is viral particles in your nose. And so since it might not have come all the way down because, you know, you may have taken a Kleenex and cleaned out that part. So they had to go a little further. That's why they went up as high as they did. They wanted to make sure they got in there.
My nose burning just thinking about it.
Oh my goodness. So yeah, so they literally took the swab to take some antigen and they put it on a surface. So just like we're putting it on a surface. So what you just did in this first step is you took the antibody in this case. So we're doing it in reverse, but you took the antibody and you're putting it onto a surface to immobilize it. So you're basically taking the antibody and allowing it to attach to the plastic. Okay, that was the first step. So I don't know how many minutes we have left. Chastity, how many minutes do you have left? 4? Okay.
Yes, 4.
And Daisy, what about you?
3.
3. Okay. Stephanie?
I have a minute and 40 seconds left.
Oh, you're almost done. Okay. Well, Stephanie, I'm going to have you we're going to wait on Chastity. Okay. What about you, Janae?
I did not set a timer because I thought we was just going by one timer, but
oh yeah, no, you have to do it. Yeah, you have to do it too. That's okay. Don't worry about it. We're waiting on Chastity, so okay. When hers is up, then we'll start on the next one because I know everybody will have 5 minutes in. That's okay. You haven't destroyed the test. I promise. Oh my goodness. So Chastity, you're at Druid Hills High School. I did my postdoc at Emory, so I remember Druid Hills well, but I don't remember, I'm trying to remember where was the high school. I remember there was a church, a big church on North Druid Hills.
Right by Emory, like literally right. Like if you walk, it would be on Emory campus.
Oh, okay, gotcha. Yeah, like I said, I remember being over there on North Druid Hill because I used to go to the Einstein Bagels on North Druid Hill. Yeah. I don't know if it's still there or not, but is it? Gotcha. And Stephanie, where are you from? Are you here in Columbus?
Yeah.
I say here in Columbus as if I'm here in Columbus and I'm not. I don't even live in Georgia. Daisy, what about you?
I'm from Columbus.
Oh, okay, you live in Columbus too. What about you, Janae?
I live in Macon, Georgia.
Oh, okay. Awesome. Awesome, awesome, awesome. Let's see, I went through Macon to get to the big city of Toomsboro, Georgia. Ever heard of it? See, yeah. I picked at my friend, my best friend, one of my best friends from college. She was from Toomsboro, Georgia. I think they had a population of 200, if that. And I think half of them were her family members. But but I drove through Macon to get there. Oh my goodness. Yeah, no, I know you're like, I don't know what that is, but that's a little bitty place. And it really is. I'm trying to think, what was the next little city that was next to it? I can't even remember the city that was next to it. So do you guys have any questions for me? Anything about my crazy life?
How do you like being a doctor? What kind of doctor are you?
So that's a good question, because I get that a lot. Some people say, hey, what kind of a doctor are you? Because I studied virology and diseases, people automatically assume I'm a physician, and I'm not at all. So I have a doctorate of philosophy, which is a PhD. And so when I finished my virology degree, I actually went to Emory and went at the Winship Cancer Institute and worked doing cancer research, which is what I still do now. And so because of the cancer research, people also think, oh, can you tell me about this? And I'm like, I don't write prescriptions or anything like that, nothing like that. I just do the research on the other side. But that's what I did. So I was there at Emory for a while. So that was pretty cool. That was a lot of fun. A lot of fun.
I'm going to school and I'm going to major in biology pre-med and ultimately to be an ER physician. So that's
okay. Awesome. Yeah, one of my, matter of fact, my friend who's the Toomsboro person, that's what she is. She's an ER physician. And she likes it a lot. She does ER, well, actually she does ER in the evenings, and she does her regular family internist practice during the day. But she really likes the ER because there's just a lot going on. And so yeah, you just never know what you're going to get. Whereas as an internist, you kind of know what you're getting, because when it's flu season, everybody's coming in for the flu. If it's strep, everybody's coming in with strep. So for her, it's like, that's cool, that's fine. But then she does like the ER, and she likes all of the changes in the ER. So that's fun for her.
Me too, because I get bored really fast. So I'm like, I have to go into something that's constantly like fast-paced. I always have something to do.
Right.
I get bored.
That's a good thing to know about yourself, and it helps when you know those things about yourself. It helps you decide what career is best for you, right? Not everybody is good with a lot of change. For some people, change is something horrible. I mean, it completely throws them off. I have a friend who was just like that. I mean, we could be doing whatever, and then just as soon as things don't go the way she had planned, and things happen. Oh my gosh, you would think the world had ended. It's like, oh my gosh, it's over. It's over. And so for her to have a job that changes a lot is not the best thing, because, that's something that she just doesn't do well with. So it's always good to know your personality and how you are, and it helps you decide which career is best for you and best suits you. Let me see. So Chastity, what do we have for time?
I didn't want to interrupt, but the timer went off.
Oh, okay. No, you could interrupt. You're good. All right. So ladies, let's move on to step number 5. So step number 5, you're going to get a new tip and put it onto the end of your pipetter. And then you're going to take your negative control. So let me see your negative control tube. Everybody put your negative control tube up to the camera. I want to make sure everybody's got the same one. Right. The white cap, right. There we go. All right. So we're going to take 50 microliters of this. And remember, I told you, you're going to have to decide which well your negative is going in, whether it's number 1, the 2s, the 3s, or the 4s. So whichever one you decided, that's the one I want you to put this negative control in. So I want you to take 50 microliters out of that tube, and I want you to put it into the first well that has that number. Then I want you to go back into that same tube, take out a second 50 and put it into the second well of the same number. So for instance, if you decided negative was going to be in well number 1, then what I want you to do is put 50 microliters into the first well that's labeled 1. And then I want you to go back to that tube, take another 50 and put it into the second well of the tube labeled number 1. When you're done with that, what I want you to do is take that tip off, put it into that cup that you got the water in, get a new tip, put it onto the end of your micropipetter. And the next tube I want you to grab is your positive control tube. So everybody hold up your positive control tube. Right, the blue. Perfect. So we're going to take 50 microliters out of there, and you're going to put it into whichever number you decided was going to be your positive. And we'll repeat the same thing again. Wait, yeah, just throw those away. We're not going to use them again, Janae. We're wasteful. Yep, get a new one. You need a new one. Oh, you only had 2 in your thing? Okay, well, go ahead and rinse that one out and rinse it out really well. And
you said throw it away. It's a trash can right next to me. I just threw it in there.
Yeah, you can take it out. We'll be okay. But you should have had 10 instead of 2, but that's okay. We'll just rinse it out with the water and then do it again. Okay. Does everybody else have like more than 2?
I can't find it. It's gone.
Okay. Yeah. See how you got the bag? Yeah, you should have a whole bag full of them.
What bag? Let me see. Oh, this. Oh, okay. I got you.
All right, now we're on. We're back on. We're back on. We're back on. So that was your positive control. And then you'll do the same thing for patient sample number 1. And what color is the cap on patient sample number 1?
Hold on, wait, I got a question.
Green.
You said do what with positive control? Because I didn't
get to do so which number did you decide to put your positive control in? So on the sheet, you were to put which one was going where.
Okay, I did. Okay.
Okay. So you'll follow that, whichever one you made the decision on. And you'll repeat it for patient sample 1 as well as patient sample 2. I kind of feel like you guys are kind of in a rhythm now. You kind of know what's going on. And when you're done putting all of those in there, what we're going to do is we're going to wait another 5 minutes. So make sure you have your cell phones or whatever you need that you're using as a timer and set it for 5 minutes. Yeah. And just make sure you change your tip each time. Yep. That's it.
I'm sorry, I'm the last one.
No, you're okay. So Stephanie, are you finished with everything or are you still working?
I'm doing the last one, patient 2.
Okay.
Yeah, me too.
All right.
All right, I'm done.
You're done with, did you do patient sample 1 and patient sample 2?
Mm-hmm.
Okay. So set your.
I'm gonna set my timer.
Set your timer. Is anyone else done or someone else still working? You're done? Okay. Daisy, are you done? Perfect. Stephanie? Okay. Excellent. So now what you've done is you've actually added your sample. And this is the unknown thing, right? We don't know what is in these things. We don't know if it's going to attach or not, okay? So remember in the first step, we attached our antigen antibody to the plastic and it was gonna stick to the plastic. Then we added our next sample and the next sample is, we don't know what that is, okay? But if you remember in the ELISA assay, remember it was like a sandwich, right? You put your antigen on or antibody and make it stick or immobilize it. Then you take the opposite, which in the example I showed you was the antibody. In your particular case, it's a sample. And what you're allowing right now in these 5 minutes is you're allowing these two samples or these two molecules to attach to one another. You're waiting for them to bind to one another. And if they recognize one another, they will bind. If they don't recognize the other one, then they won't bind at all. You'll just have two things floating around in liquid just independently of one another, okay? So right now, we're just simply waiting to see, will these two things bind to one another? Now, one thing you'll notice is as you're looking at this, you can't tell if they're binding to one another or not, can you? Because everything is just clear. You can't see anything happening, okay? And part of the ELISA is you want to be able to detect this interaction. You want to be able to see, did this actually happen? Did these two things actually bind to one another? Because I can't see in this liquid as it is if they did or not. And so when the 5 minutes are up, you'll notice that you're going to add a second thing. So in step number 10, without actually doing step number 10, what are you going to add in step number 10?
Apply a new clean tip to detection antibody.
Right. You'll see that 2 and the 0, right?
Mm-hmm.
So that means secondary. So that's our secondary detection antibody. That particular antibody is going to attach itself to those samples that you just added. The patient samples, the negative, the positive, is going to at least attempt to bind itself to those samples. And if it does, then you'll be able to see how you got this constant sandwiching one on top of the other, okay? So what is the time? What are we looking like with time?
Oops. I have 1 minute and 36 seconds.
Okay. What about you, Daisy, Stephanie?
My time is up.
Your time is up? Okay, what about you, Stephanie?
I have 1 minute and 20 seconds.
Okay, what about you, Janae?
1 minute and 36.
Okay,
No,
so we don't have long at all. Everybody's right at a minute. So it won't take us too long, but yeah, so right now you're just waiting for those samples to attach themselves to the antibody. And so what we'll do next is we'll add the detection antibody. So you're almost, I'm sure you're almost done with the timer. And when you are, when you add the detection antibody, you'll add it to all 8 wells, all 8. Okay. Make sure you add it to all 8.
So just do it when the timer goes out.
Mm-hmm.
Okay.
Yeah. How much longer do you have on your timer?
18 seconds.
Oh, okay. You don't have long at all. All right, but if you guys want to go ahead and get your secondary detection antibody, and what color is that, Cap? Purple, right. Okay, so go ahead and add 50 microliters to all 8 of your wells. And you can keep the same tip on this one. So you'll just drop it into all 8 wells. You guys are legitimately focused. And when you're done with all 8, what I want you to do is set your timer for 5 minutes again.
Oh my God. I'm sorry.
All right, is everybody done? Or is there some, okay, you're done, Stephanie. What about you, Chastity? Okay. I think Daisy is still working, I believe.
I'm done.
Okay. So now that you've added your secondary, what we're going to do now is, this is the antibody that's actually going to, in this particular case, it has a color, a molecule that's colored on it. And this particular molecule is an enzyme. And as you probably remember, enzymes and substrates interact with one another to produce a product. And so the last step, which is after this 5 minutes, you're going to add a substrate. And what's going to happen is that substrate is going to interact with that enzyme. When it does, there's going to be a color change. That color change will only happen if all of these things line up and bind to one another. So that, remember, in step number 1, we had the antigen, the primary that we attached to the plastic dish, right? Then we added a sample. And if the sample does what we hope it does, it will attach itself to the first antibody that we added. Then the third thing we added, which you just did just now, was the secondary. And so we're hoping that it will then bind to the sample. So you've got your primary that's attached to the sample, that's attached to the secondary. And then the last thing will be the substrate. So if all of these things attach to one another, you're going to see a color change. That's going to indicate a positive result, meaning that that particular sample is positive for the virus. Okay. Again, this is a simulation. This is not real. So you're not doing real viruses. I wouldn't do that. But it would be as if it were a real virus. Then what you can say is that particular sample has virus in it. Okay. And so when we get to the end and you add the substrate, and it's going to be an immediate reaction, you'll be able to tell me which of these samples has virus in it and which doesn't. okay? Or at least simulated to have virus in it, okay? All right, so while we're waiting on this and waiting on things, let's look at the questions that's on the data answer sheet. So for the first question, it says, what is the name of the virus that causes COVID-19? Anybody know the name of the virus that causes COVID-19?
SARS.
Close, that's part of it. The rest of it, the rest of the name. So the name is SARS-CoV, CO with a capital V for coronavirus 2. Okay, number 2, which is different from SARS-CoV-1. So this is SARS-CoV number 2. So that's the name of the virus that causes COVID-19. So, and we'll have to wait for numbers 2 and number 3. But there are 2 tests that are used to identify COVID-19. Anybody remember or know which 2 tests do people have if they're being tested for COVID? There are 2 different types of tests that they can have. Anybody know what the 2 tests are?
The rapid.
Right, the rapid test, which is this. This is what we're doing right now, is the rapid test. And what's the other one?
I'll let somebody else get it.
You said somebody else can answer that one. Somebody else can answer. Anybody remember what the other one is, the other type? All right, the other type is PCR. PCR is the second test, and that one takes longer. The PCR test was the first test that they utilized for testing COVID-19, but it was taking a long time because what you do is you take a sample, again, out of the nose, right? You're taking that sample, and what they were having to do was extract the RNA, DNA, or the nucleic acids, but they had to remove that, separate that out of the sample, and then do a PCR test to actually detect viral parts in that sample, i.e. the spike protein. Yes, ma'am.
My phone on 2%. I'm finna go get my charger right quick.
Okay. And so that particular test take a little bit of time. It took like at least an overnight run because you have to do multiple rounds. And so that one took a long time. It wasn't until they did the rapid test, which is what you guys are performing right now, that people were able to check and see who had what faster. Now, with PCR, you're actually detecting the presence of the viral nucleic acid. In this particular case, we're talking about the viral RNA. So we're trying to see, is the viral RNA there, which tells us that you have virus in your system. But with this particular test, we're actually looking for an antibody response. So we're just trying to see, did your body react to the virus that is present? So when it comes to advantages and disadvantages between the two, the PCR test was the most accurate. Most accurate in that particular test could actually tell you, is the actual virus in your system? Because you're actually able to isolate viral nucleic acids from that sample. The other test, the rapid test, is actually for antibodies. But as you all may suspect, you can produce antibodies to something and have them still hanging out in your body, even though the virus may have cleared and is out of your body. So what the rapid test does, it says whether or not you have been exposed to a coronavirus. It doesn't necessarily say it's in it right now. is in your body right this second. But it does tell us for sure that you've been exposed to it. Okay, and so those are like some advantages and disadvantages between the two tests. All right, so what do we look like on time? We're good?
My timer went off.
All right, so I'm sorry, what's that? Is somebody
Nothing,
still
I just
waiting?
said my timer went off.
Oh, okay, great. All right, so what we need to do now is get a new tip. And what we want to do now is take the tube that says substrate. And what color is that cap? Right, the brown one. So we're going to take that particular substance and you're going to take 50 microliters. You're going to put it in every single well, all 8 wells. And you can keep the same tip this time. All right, anybody seeing anything happening? All right, so when you're done, hold up your plate and let's see. All right, Janae, you got your cord?
Yeah, I do, but it's short. I can't. Hold on,
Oh,
I
that's
can do it
okay.
like this, though.
Okay. Yep, there you go. All right, so
Janae, hold
you
it
got
back
yours,
up.
Daisy? Oh, I'm sorry. Photo op time. You gotta hold your plates back up. All right. So now going to question number 2, it says, which patient is positive for COVID-19? So who's positive for COVID-19?
Patient 1.
Right, patient 1. Who's negative for COVID-19?
Patient 2.
Patient 2.
All right, how do you know that patient 2 is negative?
There
wasn't a change in color.
Right, there was no change in color. For those that were positive, there was an actual change in the color, correct? All right, awesome. So when you think about the patient sample, what do you think the patient sample was made of? Do you think the patient sample was antigens or do you think it was the antibody?
I think it was the what'd you say? Say it again, please.
Do you think it was the antigen or do you think it was the antibody? What do you think the patient sample was?
Oh, let me think. I think it was the antibody.
Okay. All right. So, and you're right. And so with that being the case, you can actually see whether or not somebody had an immune response to the virus. Okay. All right. So what are your thoughts so far on this? What do you think about the test? Is it pretty simple, straightforward? Do you think you have a better understanding of the COVID test now, the rapid test at least, and how it works?
Yes, I like it.
You like you like it. It doesn't take all day, right? Now you understand why with the rapid test, why they called it the rapid test and why it was so much faster. Okay? Because you get an immediate response. Whereas with the PCR, it takes a lot more fancy equipment, it's a lot more expensive. And for those of you who may know someone who did the PCR test, it costs more because of the amount of work and the reagents that are used to administer it. But with this one, you see this was pretty simple, pretty straightforward, not a lot, and therefore this particular test doesn't cost as much. It's a lot cheaper, okay? Because you don't have to worry about having fancy equipment. You just simply mix it together and you see a color change, and that's it, okay? All right, are there any questions that you guys have? Because that was the end of your lab experience. All right, well, if there is nothing else, thank you ladies so much for joining me. I hope you enjoyed the activity. I definitely enjoyed meeting you all. I wish you all the best. Janae, I wish you all the best at Savannah State, right? That's where you're at or heading to?
Yes,
all right.
ma'am.
All right, awesome. So I hope you got all your shopping in and you're done and you're ready to move in, but you probably tell me, no, you're not ready yet because you haven't done all of that yet. That's okay. That's okay. And for the rest of you ladies, enjoy your senior year. I'm excited for you guys to have like a normal senior year. I really am. So enjoy your senior year and make sure you get all of your things ready so that you'll be like Janae this time next year, ready to
Dr.
get things
Frazier,
together.
Thank you so much for everything. This was awesome, fantastic. I learned so much. I hope the girls did too. Randall, thank you for facilitating. Cheryl and Melissa, thank you for all you did to get the girls their items. Girls, thank you for being here, for being so engaged. We appreciate you, and we are going to do a round 2. So if your friends hear about it, make sure you encourage them to sign up so they can participate again as well. We're going to send all of you. Normally when we have a large group, we only do 1 door prize, but since you all signed up, you all get a door prize.
Woo!
So we'll be sending that to you very shortly. So thanks again, guys. Have a great evening.
Bye.
Bye-bye.
Dawn. Some of the girls indicated they were having some difficulty with their Grubhub. I wanted to go through and just kind of ask, was anyone not able to utilize their Grubhub?
I didn't see email.
I'm going to let Cheryl stay on and help walk them through that as well. Thank you, guys. Thank you, Dr. Frazier.
Okay, what I would suggest, we did a test. I actually sent it to one of our staff, and she had no problem with the link, and she was able to see the credit in her account. So what you have to do is, did all of you have a Grubhub account prior to this event?
Yes.
So no? Okay. If you did, you have to, what you're doing when you follow those steps on the email and memo that you received, it helps you link your account to the event. So your email address that you provided for the registration is what links you to the event. So I would suggest you log out completely out of Grubhub and log back in using your credentials. I'm thinking that your email address should link you to the credit. You may see it now. Now before 6:15, it was not visible. That's how we hold the credit until everybody signs into the meeting, and then we release it. So it is visible now.
And I just wanted to state that we used your email address that you put in when you registered. So Stephanie, yours is a Gmail. Daisy, yours is a Gmail. Janae, yours is a Gmail. Chasney, yours is your DeKalb school email. So
did all of you receive the welcome email from Grubhub? It comes directly from Grubhub. I know the email said it would come from me, but I generate the email, but it will come directly from Grubhub. And that was earlier today around the same time Miss Randallette sent you a communication. So shortly after, you received her email, you should have gotten something from Grubhub saying welcome. And then it kind of walked you through some steps to register your account. So did everybody get that email?
I didn't get a welcome email, but I got the email from Miss Miss Williams.
Okay, so Janae, you said you did not, but everyone else received it? Okay, so you followed the steps. And that should link you. Now I'm going to go in now, Janae, and see if I can resend it. So hold just a moment.
So who was it that didn't get it?
Janae
Okay.
Brown. You can log in too. Can you see Melissa?
Oh, on the back end?
Yes.
No, I was just wanted to double check that Chasney got it because she used a district email and sometimes they block stuff. You got the email?
Yeah,
Yes,
that's
I got
what
it.
Okay, good.
Okay, let me resend. Okay, Janae, I'm just double checking. The email address is JANAE Brown 6603@gmail.com.
Yeah,
That
that's
correct?
right.
Okay, I'm sending it now.
All right, thank you.
And I got the same message. Welcome. Your welcome email was successfully sent.
It just came through.
Okay. Okay. Is that Daisy Cruz? Okay, so Daisy, is this correct? DAISYMCRUZ 2004@gmail.com. Okay, that's where it went. I'm going to resend it. Okay, your welcome email was successfully sent. That's the message that I received. Do you see anything? Okay, Melissa, I'm going to step in. I'm going to jump over here to the budget and extend the time just a little bit because
Yeah.
it's 7:32. Okay.
Daisy, has it came through yet? Great, great. So with her extending the timeline, that means you guys have 30 minutes to put your order in. It doesn't have to be delivered by 8. You just have to order it. And then if it's are you doing 8 o'clock, Cheryl?
Yes.
So if it's 8:01, the credit disappears. So you need to make sure you get your order submitted by 8 p.m. Okay, so we just want to double check everybody that after you've got your email, you've done your password, you logged on, that on the main Grubhub page, you see where it says you have a line of credit for $15 for the hands-on biotech workshop.
Okay, Melissa, I'm going to make it 8:05. How about that?
I think that's good. I'm just going to share my screen really quick so you guys can know what this kind of looks like. So I have a few different types of events and stuff, but when you first log on to the home page, you can see like this is me. Hi, Melissa up here. This is what you're looking at, this line of credit. So yours will just have one. Yours should say $15 Gear Up Biotech Workshop, and I have it refreshed. So after I refresh, this will say 8:05.
I was actually gonna say 810.
It's gonna say 810.
Yes.
But yeah, it's right on this homepage, and you're looking in this area for your credit. It's called credit. Okay, so we just want to make sure that everybody at least has gotten onto the homepage, the Grubhub homepage now, and see that you do have credit to your account. Stephanie, you good? Yeah. Daisy, are you there? Okay. Well, all right.
You see your credit, Daisy? You do see your.
Thank you, Cheryl.
Okay, and what about Janae?
I guess she was good to go. She went ahead and dropped the call.
Okay.
As well as the other young lady. I guess she was good to go also, and she's gone. So I think everyone is all right. Thank y'all so much for your help. And girls, thank y'all for keeping your cameras on. Let me just get a picture of my MCSD babies by themselves. And Daisy, I still have your gift card. I haven't forgotten it. So I'll be mailing it to you. Miss Z reminded me, you need to send Daisy her card. I was like, yes, ma'am. So let me get a quick pic of y'all. Okay, thank you. Y'all enjoy your evening.
Have a nice night. Bye.
Bye. Bye.